Hi, so my group decided to focus our project on the effects of sugar on the brain, specifically in terms of addiction. So there's been a lot of talk in the media about whether or not sugar is addicting, and it turns out that there is a lot of evidence that in certain cases, sugar can produce addictive behaviors and addictive neurochemical responses in the brain. So sugar, like any other addictive substances, hijack the brain's natural reward system to release an unnaturally pleasurable response in the brain. And so this is great as an evolutionary strategy for survival, as it creates food-seeking behaviors in animals and places where food scarcity is frequent. But in an environment that we now live in, where sugar is readily available at all times, this presents problems for humans. And so first, repeated use of sugar leads to downregulation of enkephalin, which is a natural opioid, and increased binding of mu receptors. Both of these, enkephalin binds to mu receptors to release dopamine in the brain, and this is how we see that reward system work. Second, sugar disrupts the acetylcholine dopamine balance in the brain. Naturally, acetylcholine is an antagonist to dopamine, but in drug addicted and sugar addicted brains, there's excess dopamine and decreased acetylcholine, leading to extra seeking behaviors. And this also leads to withdrawal symptoms when this balance is disrupted.
And in addition to causing addiction, excess sugar consumption has harmful effects on cognition, and it's really damaging to the hippocampus in particular. Rats that were placed on high sugar diets have shown decreased neurogenesis, as well as increased inflammation and oxidative stress in the hippocampus. And as we've learned, the hippocampus is where place cells are located, so these rats showed diminished place recognition memory on tests as a result. Excess sugar consumption can also reduce the mitochondria's ability to produce energy, and this disruption in metabolic function has been linked to depression, dementia, and other neurodegenerative diseases. So much so, in fact, that Alzheimer's disease has been referred to as type 3 diabetes in the literature. High sugar diets decrease levels of brain-derived neurotrophic factor in the hippocampus, which is a protein that plays a large role in synaptic plasticity. So when researchers placed rats on these diets, they found it impossible to induce either long-term potentiation or long-term depression in the rats. And the rats exhibited a significant decline in their number of synaptic contacts, as well as a decline in cognitive function. There is hope that some of these effects are reversible, so the rats' spatial learning performance and LTP were able to recover after they were off of the unhealthy diet for a couple of months. But other factors like LTD never went back to normal. So if you're thinking of cutting back on sugar, now would be a good time to start.
So I'll be rounding out our presentation for today by talking about a study that was done earlier this year that kind of contextualizes sugar addiction in the general field of addiction psychology and neuroscience. So again, this study was done earlier this year, and it was kind of investigating what is known as the dopamine hypothesis of addiction. The dopamine hypothesis of addiction is something that that scientists have kind of come up with to explain why addiction occurs and why people continually fall into this habit of abuse. So the hypothesis states that individuals suffering from addiction have a decreased interest in non-drug related stimulus. These stimulus usually cause some kind of dopamine release in the brain, but because of their continued drug use, the dopamine response has actually become less. So they actively seek out the dopamine releasing activity of drug use. And furthermore, the hypothesis kind of states that more addictive substances release more dopamine, and that causes the difference between being addicted to something like methamphetamine, which is highly, highly addictive, versus something like ibuprofen or something which does not cause this kind of dopamine response. So in this study, the researchers had mice, and they put these mice into a box where they were able to kind of decide between two substances. The substances that they used for this were sugar water and cocaine. When the mice would choose whatever substance that they wanted to choose, they would be injected via IV with whatever substance that they were using. So this means that it was inserted directly into their bloodstream and was hypothetically able to perfuse their system fairly quickly. So again, this first case scenario, they had just sugar water and cocaine and allowed those to naturally perfuse into the body. And in this case, as you can see on the graph that I'm sharing, you can see on the most left-hand side, the pink circle, or not the pink circle, the left circle that has a lot of the bright blue. You can see that 80% of the mice actually decided to choose the sugar water, which we might find surprising because we do consider cocaine to be a highly addictive substance and sugar water to be not necessarily something that we think of as an addictive substance. So the other graphs show some delays in giving the mouse whatever IV injection that they choose. And the importance in this is that the researchers kind of found that it wasn't so much the dopamine hypothesis and the amount of dopamine that was released that caused the choice between these two addictive substances, but it was actually the delay in response that came after them being injected. So for example, in the first case, on the most left-hand side, they found that through biometric studies, they found that it took a lot longer for the cocaine to actually get into the mouse system and for them to experience that high. Whereas when they were able to put sugar water in, they were able to feel the repercussions of that much quicker. So they were able to have a higher blood glucose level a lot more quickly, and their body generally felt that effect faster, which meant that hypothetically there was dopamine released quicker. So they hypothesized then at this point that what was actually responsible for these preferences was the delay in response from the drug. So in order to further test that, they manipulated the studies such that in the middle case, cocaine and the sweet water had the same amount of delay in response time within the body. And in the furthermost on the right side, they made it such that the cocaine had a much shorter response compared to the sugar water. And they just did this by changing the time in which they injected the substance after the mouse made a choice. So meaning they would inject later to change the manipulate the times. So again, if we look at this as a whole, we can see on the left-hand side, the mice chose the sweet water more often because there was a smaller delay in physiological response. In the middle, when they made the delay about the same, and just for context, cocaine does take about 30 seconds for it to cause some effect in the mice. So when they were able to make those response times about the same, there was about a 50-50 choice or chance that the mice would choose either one of these substances. And then on the most right-hand side, we can see in this case, they made the sugar water have a much longer response time than the cocaine. And in that case, the mice actually switched their preference for the substance that had a shorter response time. So in this case, they started to choose the cocaine, which impacted their body more quickly. And as a whole, hopefully this makes sense, but this was a really kind of genius study to test these hypotheses and kind of show how much our understanding of neuroscience changes and how simple these studies can really be, but still provide us with all this amazing contextual information. So thank you for listening to our presentation. Thank you all for a great semester, and sorry I couldn't be there today, but thank you all. Bye.